Hi everyone, thank you for joining us today for the bioCADDIE external webinar. Today's speaker is Dr. Mark Musen, and he is a professor of biomedical informatics and of biomedical data science at Stanford University, where he directs and is the principal investigator for the Center for Expanded Data Annotation and Retrieval, CEDAR. And this CEDAR is a center of excellence that is supported by the NIH Big Data to Knowledge Initiative and has the goal of developing new technology to ease the authoring and management of biomedical experimental metadata. And if everyone could please put your microphones on mute unless you're asking a question, that way we won't have any background noise during the presentation. So Dr. Musen, please feel free to go ahead and get started.
Well, thanks so much for that introduction, and thanks to everyone for dialing in. What I'm going to talk about today is the Center for Expanded Data Annotation and Retrieval. I'll give you an overview of the center, some of the things that we're doing, and obviously the motivation for why we've started this program and where we think it's all going to head in the long term. What I want to do is first, though, sort of give you some background as to where we're coming from and what we're trying to achieve, and then we'll talk in some detail about what CEDAR is all about. Like many of you, when I was in college and earlier in my career, I always turned to the Journal of Irreproducible Results as a source of humor, and always thought it was funny to read these kinds of articles. What's happened to science in the past 4 or 5 years, though, has been really a change, where I think to a certain extent the public has become increasingly concerned about science and the reproducibility of results and the veracity of what scientists do. And as a consequence, I think all of us in the scientific community have tried to respond. We've been stimulated not only by the public's concerns, but what we have read in journals. Probably the most infamous paper was by Begley and Ellis a few years ago, where they described Amgen's attempt to reproduce the findings in 53 papers that they considered landmark papers in cancer biology. And of those 53 papers, they could reproduce the findings in only 6. And they cited a study by Bayer that could validate only 25% of some preclinical studies, and recognized that a lot of these landmark studies, which people could not reproduce, had spawned entire fields of investigation where the original results were assumed to be obviously correct, and where everything was dependent on those original results that it turned out were difficult or impossible to validate. And Begley and Ellis also pointed out that the non-reproducible studies were most likely to be published in the journals with the highest impact factors, because that's obviously where most people wanted to put such results. And as you know, there's been a backlash, there's been a lot of hand-wringing, and NIH has tried to respond in many ways, recognizing that this problem of confidence in science has lots of causes. There's lack of statistical power. There is clearly an art form in conducting experiments, and it's sort of like baking a cake. Some people can just do it well, even though you think the recipes are the same. There's also an eagerness to publish early. There's obviously a certain amount of outright fraud. But we also have a system that doesn't make it easy to reward or rewarding to replicate the results of other investigators. And we certainly have a system where it is not easy to make your results so explicit that others can easily replicate your results. And I think at a minimum, science needs to have open online access to experimental data sets that people can look at, they can examine, they can validate. There needs to be annotation of online data sets with adequate metadata, the data about the data, and that of course is what CEDAR is all about, and I'll get there in a moment. Those metadata just can't say what's in the data set. They have to use controlled terms that have explicit meanings. There have to be mechanisms to search for the data, to find the relevant experiment results. And fundamentally, what this all means is that we need to be able to use metadata to make sense of the experimental results that people have achieved. And not only do we need metadata, but we need metadata that are based on standards. Standards for the kinds of things we want to say about the data, for the language that we want to use to say those things and how we structure the metadata. So that's fundamentally what CEDAR is about, and I'll get, as I'll say. I would also add that we need a scientific culture that values all this stuff. And although CEDAR has no expectations of creating cultural change, we certainly hope that the technology we're developing will change to a certain degree the ease with which metadata can be authored, and thereby have an effect on changing the way that scientists may view the creation of the metadata needed to describe their results. So let me tell you a story that you probably are familiar with. Some 16 years ago, when people started to think about DNA microarrays and the high throughput results that came from those kinds of experiments, we were in a situation, or the microarray community was in a situation, where they recognized that fundamentally there were problems in being able to describe what was in a microarray result. And it was also recognized that obviously when you had an experiment that resulted in information about 25,000 genes over several time points, you couldn't exactly publish that in a paper-based journal, and all of that would have to be put online. But it was impossible to understand what people had done in a microarray experiment unless there was information provided about what was the substrate of the experiment, what was the assay platform that was used, what was the manufacturer, what was the lot number, what were the experimental conditions under which the experiment was done. And what that led to was not anything that came from NIH, not anything that came from the journals initially, but what really something that came out of the microarray community itself, where they recognized that what they needed was a minimal information set that would be ascribed to any microarray experiment in the metadata that were published online with the experimental results, so that people could actually make sense of the data. And the investigators involved in DNA microarrays at that time created Miami, the minimal information about a microarray experiment. And Miami, as published in a journal, was basically a checklist. These are the kinds of things that you need to say about a microarray in order for someone to really understand what you did. You have to describe the raw data, you have to describe how the experiment was achieved, and it was basically a checklist of everything that needed to be incorporated into the metadata that would describe microarray experiments. And Miami was really a breakthrough, because what it did was to provide a structure for metadata for online repositories. And very quickly, the Gene Expression Omnibus, which is the NCBI repository for microarray data, and Array Express, the EBI repository in Europe, both adopted microarray metadata that were based on the Miami structure. And Miami provides a framework basically for ensuring that the scientists can really understand the basics of what was done. And what was, I think, really interesting is that lots of other groups interested in other kinds of experiments have realized that having minimal information about their particular kinds of investigations can be very valuable. And so many of you are already familiar with the biosharing resource that Susanna Sansoni's group maintains at Oxford. If you go to biosharing, you'll find lots of information, including information about the Miami standard, which is available in a very convenient web-based format, and also about hundreds of other guidelines that are like Miami, that are valuable for describing the kinds of metadata that need to be included in the descriptions of various kinds of experiments. So the good news is that the biological community has recognized that the reproducibility of science, the understandability of science, all require good metadata. And many groups of investigators have gone to work creating these minimal information checklists, and they're just being advanced throughout the biomedical community. The problem is that a lot of investigators, I would say most investigators, still don't view these minimal information checklists as minimal. They view them as rather burdensome. And there's really a pervasive problem of, what's in it for me? Why should a particular investigator go through the effort of filling out the microarray description, given the complexity that they perceive is embodied in all the things that the checklist requires, and given the fact that from their perspective, their work is done when they publish their results in a scientific journal, putting the data online, creating the metadata for those data, that's just viewed as a lot of extra work. And indeed, it is a lot of work. And so when you think about what it means to take a microarray result and put it into Geo, it's more than just creating the metadata. Investigators have to get the summary data matrix that comes out of the instrument. They have to package up their raw data file, create a zip file, submit that to Geo, and wait and see whether they've actually done it right to see whether their result is accepted. And there's obviously difficulty in doing it right, because when you submit to Geo, you have a spreadsheet that you fill out, but the spreadsheet is incredibly complicated. And the spreadsheet doesn't really provide much guidance in terms of how information gets filled out. And indeed, if you look at some of the results that are in Geo, you see that people have really done not such great work in defining the metadata for microarray experiments, because when you look at the results, you see incredible ambiguity and errors. Just our analysis of some Geo results shows that if you just look at the fields that people create for age, they specify age in a gazillion different ways, which means that if you want to be able to find particular experiments on subjects of a particular age range, then the reliance on free text in Geo makes it really hard to get the information that you need. And so we have this problem where creating the metadata is difficult, assuring its consistency and its quality is difficult, and as a consequence, investigators can't really rely on the metadata that are associated with public data sets to provide the guidance that they need in order to be able to access experimental results that other people have done, to reinterpret those results, to combine their data with other people's data. And so there's really a need to make it easier to author metadata, to make those metadata more comprehensive, and to use control terms from ontologies that will make those metadata more consistent. So that's really what CEDAR is all about. We are one of the centers funded by the BD2K program, and I gratefully acknowledge their financial support. And our goal is to create the computational infrastructure to make it easy for investigators to do a good job of creating metadata so that their online data sets become fundamentally more valuable and more useful. CEDAR is a center, and we have lots of partners. I want to acknowledge not only our collaborators here at Stanford, but also those at the Oxford eScience Center, Yale, Northrop Grumman, and we also have a great collaboration with Stanford University Libraries, which I'll talk about in a moment. Our goal in CEDAR is to be able to take the online information, such as those checklists that are stored in biosharing, the ones I mentioned earlier, this slide from Susanna Sansoni shows how our goal is to be able to understand what are the elements of those checklists and to be able to translate those ultimately into templates. Templates that are not just checklists, but actually provide a framework where we can actually fill in the blanks and specify values, and to do it in a way where we have the ability to trace provenance and the ability to ensure that those templates are filled out as completely as possible and as consistently as possible. And so a lot of our collaboration takes place with the Oxford group. We also, as I mentioned, have a great collaboration with colleagues at Yale. So our colleagues at Yale are involved in the Human Immunology Project Consortium, which is a large NIAID funded group of investigators who are all involved in, well, as you would imagine, human immunology. And what's interesting about the Human Immunology Project Consortium, or HIPSI, is that the investigators here really have religion about why it's important to be able to make experimental results publicly available, why it's important to annotate them with high-quality metadata, and more than just checklists, but really real computational data structures that will provide insight into the experiments they perform. And so we're collaborating with HIPSI to be able to translate their metadata standards into a framework that can be incorporated within CEDAR, as I'll show. And we're also very excited because the HIPSI folks are very eager to use our technology and to help us evaluate it. So that's really an important component of our collaboration. I mentioned that the HIPSI folks want to make their data available online. They want to make those data online in a system called Import, which is a data repository that's been under development now for almost 10 years, funded by NIAID. Import basically is the designated repository for all experimental results that are generated by investigators who are funded by the Division of Allergy, infection and transplantation within NIAID. And other folks are actually using import as well. So it's more than just this one division within this one institute that is involved in testing and evaluating the utility of this repository. But the import folks are also collaborators with us, and they are the recipients of all of the data that come out of HPSI, as well as data that come out of many other groups. And so what this gives CEDAR, if you will, is kind of an ecosystem where we have collaborations with the HPSI investigators who are performing experiments in human immunology. The HPSI Standards Working Group, like the biosharing group, is concerned with translating checklists and other kinds of existing standards and creating new standards and translating them into metadata templates that can be used actually to acquire values that will annotate experimental data in a uniform and comprehensive way. We have the import repository that's storing all the HPSI data and the metadata associated with those data. And what CEDAR wants to do is to basically insert itself into this mix and help with the creation and management of metadata templates, to make it easy for investigators to use those templates to author metadata for import, and to analyze those existing metadata in a way which we think will inform the authoring of new metadata, and hopefully make it easier for investigators to be able to create good metadata to annotate their data and to get those data and metadata online. So here's a cartoon that kind of shows the components of what we see in CEDAR. We have a mechanism over here on the left to author metadata templates, where we are able to take information such as that comes out of the HPSI Standards Working Group, it comes out of biosharing, and to create metadata templates that will give us the ability to actually define in very explicit ways what metadata need to look like. We can then have a facility within CEDAR which allows us to instantiate those templates, to fill them in and annotate them, so that we actually can acquire the metadata that we would associate with particular online data sets. And then CEDAR ultimately will put those data sets in a local metadata repository that we'll use for our purposes. We'll also take those metadata, along with the data that those metadata refer to, and put them in the online repositories that ultimately investigators care about. Online repositories like import, as I said, but also the NCBI repositories like GEO and many others. And at the same time, because we'll have the data in our local metadata repository, we can search and identify patterns in these metadata, and as we'll see in a moment, perhaps use these metadata that we store locally to go back in our original situation where we can inform the instantiation of the metadata templates in a way which will make it easier to fill those templates out. So we believe by having the metadata in a local repository that we can examine, we can use predictive data entry to make it easier to fill in metadata and ultimately make it easier for investigators to create the metadata that they need for their experiments. So CEDAR technology is going to give us mechanisms, we believe, that will allow us to author metadata templates in a coherent way, to assemble composite templates that come out of initiatives such as biosharing in a way that reflects community standards into the ultimate templates that people will use for metadata, to fill them out to encode experimental metadata in a way which is really useful to the community. And by creating this repository that we'll use locally, we can learn the patterns of the metadata to help us in the metadata acquisition phase and predict how investigators may want to fill out templates in ways which will make it easier for them to do so. Also, I should mention that because CEDAR technology is linked to the National Center for Biomedical Ontology, which maintains the world's largest repository of biomedical ontologies, we can ensure that when investigators fill out the templates that CEDAR has in its local library, they'll be able to do that using standardized terms that are encoded using the appropriate ontology entities. And so that really is, I think, an important component of what CEDAR intends to do. So let me just give you a sense for how we go from authoring the templates to filling in the templates to using the metadata in our repository. And let me start with some screenshots of the current version of the CEDAR system. So when you log into CEDAR, you get access to a variety of different kinds of elements, including folders that contain groups of templates or individual templates. We might say, for example, that we're interested in the biosample human template, which allows us to find metadata for experimental data sets that are going to be submitted to the NCBI biosample system. And we could click on biosample human, and we can, for example, get a drop-down list which will give us the option of either populating the template or editing it or getting information about it. Let's suppose we want to populate it. Well, this is what the template looks like. So we have, sample name, an organism, a tissue, the sex of the organism, information about the isolate, the age, and biomaterials, and so on. Basically, this provides the framework for specifying the metadata that we want to upload into biosample. And this template would be filled out by any investigator who has experimental results created by some investigation where the data need to be put into biosample. And what we can do is then take the template, and let me just show you where the template came from first. The template designer in the CEDAR workbench allows us to say, for example, sample name, that text field you saw a moment ago, needs to be defined using text. The organism needs to be defined using text. Something like the age will be defined using an integer or a range of integers. And fundamentally, we have the ability to say that for anything that is defined by text, whether we actually want to associate the values not just with arbitrary text, but whether we want to actually use some ontology to specify those values, whether there might be multiple cardinality, whether it might be a required element or an optional element of the template, and so on. And so what we can do is define the template this way. We can say, for example, for the element of the template called tissue, we might say that we want to associate the tissue with a particular value from an ontology, and so we can do a search. We can say, within the CEDAR framework, we're interested in ontologies that talk about tissues, and what CEDAR will return is a list of all the ontologies in BioPortal where there's a class called tissue, or tissue appears in the name of some class, and investigators can say, what ontology do I want to use? Do I want to use Uberon? Do I want to use the mouse anatomy ontology? Do I want to use NIF standard? And basically one has the ability to make a decision on which ontology to use. We can say, for example, well, let's suppose we want to use Uberon. We can then view the Uberon anatomy ontology, get a sense for what kinds of tissues are included in Uberon, and see if this is the ontology we want to use to provide the basis by which investigators will select terms to put into that template slot that we call tissue. So what we're doing in the template designer is saying we want to associate this particular metadata slot with this particular portion of some ontology, and that provides a set of control terms that investigators will use in order to be able to specify what goes into that particular slot. So we've talked about how we author the metadata templates. Now let's talk about how we actually fill them in. So here again is that biosample human template that I showed a moment ago. And I think one of the interesting things that we're doing in CEDAR is that we are not only allowing people to fill in this template either with free text or with drop-down lists that select ontology terms, but we're also making it possible to use the database of metadata that I indicated earlier as something that we can either store for later use, but also we index in a way which allows us in real time, when one is filling out a metadata template, to get values that are recommended that might be appropriate for those particular template slots that we're filling in. So to make that clear, let me just go back to the biosample human template. And let's say, for example, that we're interested in filling in the tissue. Well, as I said earlier, the designer of the template might say that the best way to select a tissue is to choose one of the tissue types that are available in the Uberon ontology. And so the drop-down list gives us choices such as lung or colon, brain, skin, serum, abdomen, connective tissue, and a variety of others. And you'll notice next to each selection, reaffirms that the selection is coming from the Uberon ontology, so we know that it's a controlled term. We know that it comes from an ontology, so we're not going to get it wrong. We're not going to have a typo. We're not going to choose something that's not appropriate. And you see a number next to that value. That value actually tells us how frequently is that particular ontology term used in the templates that we've stored in our local repository of metadata, and how frequently do we see that particular term, so we can get a sense for how commonly used terms are, and that might help us select the best one. In the situation here, where we have a sample name and we know that the organism is Homo sapiens, which is always going to be the case for the biosample human template, we don't have much guidance. We see that there's this broad, uniform distribution of terms that come in out of our database, and so that's not terribly helpful. On the other hand, if we were to say, for example, that the tissue type is brain, as we see in this slide, then when we go and select the value for something such as disease, which is coming out of the disease ontology, then we can see where that being able to do predictive data entry is very helpful. Here, what we're doing is providing a drop-down list where we're showing the frequency with which various kinds of diseases appear in our local database of metadata in the context of templates that have been filled out using information that we have already, including the fact that the tissue type is brain. And what we see in the drop-down list, we're not seeing all gazillion types of diseases that might be found in the disease ontology, but rather just those that relate to the brain or the nervous system. And I think it's very interesting to see, for example, that in our current database, 39% of our templates that are filled in deal with Parkinson's disease in the context of brain tissues, and 27% deal with central nervous system lymphoma, which is probably not a normal distribution of diseases, but we get a sense for what the local metadata are, and this kind of a drop-down list can be helpful in making it easy for an investigator to quickly select a disease and to do it in a way which allows one to relate that disease selection to an ontology where we know that the term is going to be predefined and going to be standardized. Similarly, if we decide, for example, the tissue type is lung, well, then you see by scanning our database, we come up with very different selections. We see that in 61%, the element of the disease ontology that appears in the database is lung cancer. And although you may say, well, lung cancer is pretty broad, it's not very specific in terms of tissue type, this happens to be the way our database is formed, and we know that as we obtain more metadata, that the database will presumably fill itself out and we'll be able to get more precise terms in more frequency that will make this drop-down list even more valuable. And you've seen associated with lung are diseases such as lung cancer and chronic obstructive pulmonary disease and squamous cell carcinoma and idiopathic pulmonary fibrosis and lung cancer and so on. So we have a system where we can then fill in these diseases, or rather fill in these template slots. Here we've selected a tissue type skin of body, and we can fill in optional attributes, and we can say basically that we want to use standardized templates and standardized ontologies to make this all possible. And so we have a system whereby we can create metadata templates from these community-based standards. We can annotate the templates with appropriate metadata, and our local metadata repository allows us the ability to make the metadata acquisition itself a bit easier by providing these structured and standardized and dynamic drop-down lists. The other thing that we imagine in the future, which we obviously have not begun to implement or to really design, is the idea that this metadata repository itself may be valuable as a resource. We can use it to look at trends in scientific investigation. We can use the metadata repository as the basis for understanding what particular groups of investigators might be working on. We can use it in ways which we think will be interesting to the scientific community. And so it's valuable, I think, not only as a resource for making the metadata acquisition task easier, but something which may be useful to the community overall. One thing I really need to stress is that what you just saw in these slides showing the CEDAR features is that these are user interfaces that are generated dynamically. All the semantic components in CEDAR, the template elements, the templates as they get filled in, the ontologies, the value sets, all of them are managed as first-class semantic entities. So as you can imagine, the drop-down menus and the user interfaces are not hard-coded, but they get generated on the fly based on the semantic content. So when a user indicates he or she wants to use a particular template, we then take that template out of the library and we generate the appropriate interface for acquiring instances of that template. The drop-down menus are generated dynamically based on our search of the metadata repository to find out what are the ways in which terms are used in the particular context of the investigation being described and so on. Although I'm not going to talk about it in this presentation, our software components within CEDAR all have well-defined APIs, and one of the things that we're also very interested in being able to do is offer CEDAR not as a monolithic piece of software that runs on some web server out there, but as a set of resources that can be accessed by a variety of clients, where we believe there may be people who want to use pieces of CEDAR and be able to access those through separate APIs. One thing I should also emphasize is that all of the metadata that we generate in CEDAR is initially stored as JSON-LD, which is a widely adopted web standard. It can be translated into lots of other representations, and so we have the ability to standardize our metadata in a way which we think is important to us. As I mentioned, we're heavily involved in working with import to be able to use CEDAR metadata to annotate the HPSI data sets that are going into import. But we also have a number of other collaborations that are going on which we think will really test us in our ability to evaluate CEDAR in a variety of settings. So for example, we have an excellent collaboration with the LINKS project, which is looking at cell signaling. And we're working with them to see how CEDAR can provide a front end to the acquisition of metadata for the variety of experiments being done by their network of investigators. We are working with our local university data repository, which is based on a system called Hydra in a Box. I won't actually claim that I have any great admiration for that name, but Hydra is a system that has been developed by a large consortium of research universities in the U.S. with the goal of providing a mechanism for storing locally research data that investigators might want to make available online that wouldn't otherwise be accessible in a public repository. And we're working closely with our local folks at Stanford to see how we can work to make CEDAR an effective front end for the Hydra consortium. I guess the mantra for all of BD2K in the past couple of years has been one of trying to make data that are available online findable, accessible, interoperable, and reusable. And this notion of fair data has been extremely important to the BD2K community and I think to the scientific community overall. And although the notion of fair data I think has been widely embraced, knowing exactly how to determine whether data are fair is not exactly easy. But I think one of the things that we want to do in CEDAR is provide mechanisms where we believe that we can make online data sets more fair, if you will. And so how can we do that? Well, we believe that CEDAR is going to make data that are online more findable. They're going to be findable because not only will the data sets be associated with metadata, but those metadata will be based on standard templates that are derived from community-based standards, where we can be clear about what those metadata need to look like and can be confident that as people fill in those templates, they're filling in the templates that the community has identified as providing the kinds of slots that provide an adequate description of the experiment. We're going to use predictive metadata entry, as I've said, to encourage the creation of more complete metadata specifications. And one of our goals is that we will be able to identify the degree to which having these adaptive drop-down menus, having a predictive filling in of the blanks, so to speak, will ease and accelerate the way in which metadata specifications are created. We are, as I said, using standard ontologies and value sets to fill in those blanks, which are themselves based on FAIR principles. And metadata and the data sets they reference will all be given unique identifiers, which again makes them more findable. We believe the data will be more accessible because everything in our data sets will have unique identifiers. And the CEDAR metadata archive itself will be persistent, even if for some reason target metadata repositories are ever decommissioned. So we believe that we can enhance accessibility by our own desire to make our metadata persistent. The metadata will be interoperable because we're using JSON-LD as a standard representation language, which the world seems to be flocking to. And we're taking advantage of community-based content standards for templates, terminologies, and value sets. And so by hopefully using the structures that the communities themselves are standardizing, CEDAR will ensure that the metadata from one study will be compatible with the metadata from another. We believe the metadata that are being authored through CEDAR will become more reusable because the metadata will include provenance information for the data sets, for the metadata, and that they'll become more comprehensive because by making the metadata easier to author, we have this conviction that scientists will be able to do a better job and will be less burdened by doing their work to make the metadata as complete as possible. We also are hopeful that our adoption of community-based standards will resonate with investigators and that they will feel comfortable using CEDAR because of its adoption of the standards that the communities themselves are promoting. I realize a lot of that is promissory, and really I'm hoping that over the next year, as CEDAR becomes more robust, we'll be able to do the kinds of experiments where we'll be able to quantify the degree to which CEDAR makes data more fair, the degree to which data are more, or metadata rather, are more complete and comprehensive, and the degree to which basically we can accelerate the time it takes to create these data sets online. What's very exciting for us, and this is all obviously thinking many years down the road, is the idea that CEDAR provides a very preliminary step toward the movement that we see in the scientific community to be able to make metadata available online and ultimately to make metadata, if you will, the definitive publication framework for scientific knowledge. And I think one of the things that we want to experiment with in CEDAR over the next decade is the idea that we can increasingly formalize scientific knowledge in terms of ontologies. That those ontologies can standardize scientific communication in a way where in the long term we can disseminate scientific results in a way which is machine interpretable. And not only will we imagine scientists who want to search the literature and put data together, but we can have our intelligent agents that will allow us to access scientific knowledge and to be able to help us understand what has been done in scientific work. So what's very exciting to me, and this is obviously many years down the road, but imagine, if you will, a Siri-like intelligent agent that can tell you when a new study has been done in a scientific area that you care about, that can help you compare and contrast the results of scientific studies across various dimensions that matter to you, and can help you understand how your data might relate to the data that were done by other investigators, and help you understand how the methods that you may have used in some way might vary from those that are used by other investigators. Imagine this agent that can identify the studies whose particular subjects best match the characteristics of particular patients. So you might be a clinician who wants to be able to find a clinical trial whose subjects best match the patient whose therapy you might be uncertain about. That would obviously be a very exciting kind of possibility. And the ability to tell investigators when the methods of their proposed study are nearly identical to those of earlier studies, and in fact, where you might be able to identify when a study that you have done might actually have been done by someone else in the past. So one of the things that we dream about is the idea of CEDAR being the first step toward a technology that will allow investigators to publish their scientific results online. And the idea that one day we will have intelligent agents that will be able to search and read the literature, and I use the word literature in quotes because I think published articles may continue to diminish in their importance as what's available online ascends and takes more importance because it provides the machine interpretable representation of the scientific investigation and scientific results. These agents will integrate information and will be able to track scientific advances, hopefully even suggest the next experiments to perform, and if you believe in what people are now doing in robotics, maybe these agents could even do those experiments for us. So that's sort of where the big picture is, and I'm just incredibly excited about where CEDAR has gone over the past couple of years. Very grateful for the very hard work of the team at Stanford and our collaborators at Oxford, Yale, Northrop Grumman, and through Stanford University Libraries. Hope that the next time we get together in one of these webinars, we'll be able to tell you more information about the use of this approach by actual investigators and the degree to which CEDAR is accelerating and improving the authoring of metadata in an attempt to make online data sets more useful. Let me stop there and thank you and entertain any questions that you have. I already see some questions in the chat, and yes, the slides will be available. Any other comments or questions that folks have?
Hi, I've got 3 questions for you. The first one being, you mentioned well-defined APIs. Do you have a URL for the API docs that's either that you could point to in the slides, or that you could just let us know what it is?
Sure, I can provide that additional information. I will add that to the slide deck and make sure that the bioCADDIE folks distribute that.
Great, thank you. My second question would be for JSON LD metadata. Do you have a validation suite or something else to? JSON LD seems to be a very broad standardization format. I'm just wondering, is there something that can be used for confirming that a tool produces something that CEDAR is able to interoperate with?
We don't have that yet, to my knowledge. I'm actually turning to my development team, who is actually sitting in the room right here.
So CEDAR, when you define a template, a metadata template in CEDAR, we use JSON schema to construct that template. So the instances can be validated against that schema. So actually, validation is inbuilt to CEDAR in a sense. We use JSON schema to basically construct a template. So any conforming metadata must conform to that template. So I guess yes, the answer is yes, we do.
The answer in that sense, yes. We don't have the technology that right now interconverts between CEDAR representation and that used by other kinds of tools, but that would be coming.
Great, thank you. And last question would be, so in terms of translations or provenance information, could you share any thoughts you folks have about incorporating probability scores into the attribution of either metadata to a tool or a person or a translation process?
That's a great question, which we actually have not really considered. We're hopeful as we move forward that we'll be adopting provenance frameworks that have been used in the community, but those are all categorical, as you know. And yep. we have not provided we haven't thought about really providing a measure for uncertainty. That's an interesting idea, and I think we should consider it. But there's also the question of the degree to which we can remain consistent with existing standards.
Yeah, it is early stages for something like that, but it seems like it might be useful to at least consider the idea in the beginning rather than trying to retrofit, assuming this takes off in a few years.
Right. I mean, and particularly since we may be in a situation in CEDAR where we ultimately may want to suggest possible values for slots that are inferred from entries in the template that may or may not be explicitly evaluated and confirmed by the investigator, in which case, obviously, being able to express those in probabilistic terms would be very important.
Thank you again.
Sure.
Are there any more questions? Okay. So there are no more questions. Well, thank you so much, Dr. Musen. And the next external webinar will be January the 12th. So see you guys then. Thank you.
Thank you.
Thanks.